Bismillahirrahmanirrahim. We start another video, which is of course an amendment to the video 4, in which the first 3 questions are probably do not have any sound, and there must be some mistake I must have made. So I'm making a new video on that. So this is an amendment to the video 4, in which some questions do not have any sound. Now, if you look at question number 1, which is a slightly difficult question, as in the exam report, it says which set of measurements are correct? Diameter of capillary is given. Diameter of red blood cell is given, and thickness of the cell membrane is given. Now, if you look at the cell membrane, which is a phospholipid bilayer, now that thickness, everybody's clear, is in nanometers, and it is 7 nanometer. So then we have all these 3 answers which could be correct. Now we've got to be very clear about the diameter of the capillary and the diameter of the red blood cell. Now this is something very factual. There's no sort of ambiguity about it, whether B, C, or D, or A is correct. The answer A is correct, but why is it correct? This is what we've got to understand. The others, of course, all the figures are wrong. Now this is a very good in which you can see how the diameter is changing. So the aorta is 20 to 25 millimeter, then 5 to 10, and then 10 to 50 micrometer. And then capillary is 5 to 10 micrometer. Venule then becomes a little larger, 10 to 20, and then this becomes 5 to 20. You see, so it's increasing, and 25 to 30. I also want you to look at all these and remember all these when you're taking the for the MCQ, because you've got to know which ones have where the smooth muscles are going to be more. So they're more smooth muscle in the arteries and less in the arterioles. And then you've got to know the difference between the muscular artery would have, of course, more smooth muscle in it. Then when you look at a capillary, the size of the red blood cell is just as big as the diameter of the capillary. And this is a very good diagram. I mean, it explains it very well. There's a basement membrane on which the endothelial cells are resting, and there are intercellular clefts and where, fluid is going to be moving out. And there's some capillaries which do not have so many pores in them. So those are called continuous capillaries, and they're the least permeable. Now, another quick revision of what are the pressures in the capillary. Now, you can see here at the arterial end, the pressure is 10 millimeters of mercury. In the mid capillary, it's 0 millimeters of mercury. Why? Because here the fluid is exit the capillary, since the capillary hydrostatic pressure is greater than the blood colloidal osmotic pressure. And then there is no net movement of fluid, since capillary hydrostatic pressure is equal to the blood. Now, here the fluid enters the capillary. Why? Because here now you can see there is negative 7 millimeters of mercury. So how does the pressure change? I want you to be looking at that. I have a feeling that there might be a question on this. So this is what I want you to pause the video here and have a look at it and see if you've understood all this. So now you can see why the answer is A, because diameter of the capillary is 7 micrometer, and diameter of the red blood cell is also 7 micrometer, and thickness of the cell membrane is 7 nanometers. Then question 6, which row is correct for structures found in eukaryotic cells? Now eukaryotic cells are animal cells, plant cell, and fungal cells, and of course protozoas as well, and protoctista as well. So animal, plant, fungus, and protoctista. If you have done, if you are in A2, you would understand this. So eukaryotic cells would have circular DNA, and everybody would think, no, they don't have circular DNA. Why? Because you think of an animal cell, you think of a nucleus, and you think, well, that's got linear DNA in it. But then you don't remember that there are mitochondria present, and in plant cells there are chloroplasts present. So you got to understand is chloroplast and mitochondria. You forget which row is correct for structures found in the eukaryotic cells. Circular DNA would be present in the mitochondria and the chloroplasts. They would be present in the mitochondria and in the chloroplasts. Right? 70S ribosomes. Yes, you would say, well, they're not 70S. Yes, in the cytoplasm there are 80S ribosomes. Even the fixed ribosomes are 80S. But the 70S ribosomes are in the mitochondria and the chloroplast. So that is again something which we need to remember. Then the 80S ribosomes are of course present in the cytoplasm and in the plant cell, animal cells, and in the fungus cells. So that is why the answer to this was A. Now coming to question number 14, the enzyme beta-galactosidase can catalyze the hydrolysis of 4 substrates with similar structures. Each substrate gives a different Km value. For which substrate does this enzyme beta-galactosidase have the highest affinity? So we have to realize this is an inverse relationship. Higher the affinity, lower the Km. So let's understand this first. Now, these are the important conclusions about Michaelis-Menten kinetics. A small Km, what is a small Km? You can see here this is a small Km, and here you can see a large Km. So Km1, enzyme 1 has a small Km, and enzyme 2 has a large Km. So numerically small Km reflects a higher affinity of the enzyme for the substrate because a low concentration of the substrate is needed to half saturate the enzyme, that is, reach a velocity that is half Vmax. A large Km, like here in enzyme 2 is a large Km, is reflects a low affinity. So it's sort of inverse. High Km, large Km, low affinity of enzyme for substrate because a high concentration of the substrate is needed to half saturate the enzyme. So this is something which you need to understand, and please pause the video here and have a look at this and read all this and then figure out what the answer is going to be of the MCQ. So you look at all these: 4 into 10 raised to the power minus 3, 1 into 10 raised to the power minus 3, 2 into 10 raised to the power minus 4, and 1 into 10 raised to the power minus 4. So you know that this is the lowest figure. And so the different substrates, you had to just look at the Km in mole dm cube. And that was the smallest number. So that would give you the substrate with the highest affinity. That completes this amendment of the video number 4, in which some of the sound is missing in the first few minutes of the video. So, and I hope this is helpful to you, and please do revise, and wish you all the very best. Thank you very much.